All right, everybody, this is topic 2.4 in AP Bio Unit 2. We're going to talk about membranes and really focus in on the membrane as an organelle because um, membranes are really responsible for like defining what a cell and many organelles are. Without these membranes, we kind of have a little bit of a mess going on in the cell, or we don't even have cells at all. So, um, every cell, whether it's prokaryotic or eukaryotic, has an outer plasma membrane that separates them from their surroundings. So we see an internal and external environment. So we have internal and external. And the idea is to keep this area in here separated from the external environment, and we want to have very tightly controlled chemical environments. So we want to be very, very tightly controlled chemically environments. And I kind of compare this to like cooking in your kitchen. If you were making like a dinner with 3 or 4 different dishes, you would keep those sort of all in their own pot, pan, whatever. If you just mix the whole meal together, and I know there are crock pot meals like that, but we're thinking about completely separate dishes. Like, I don't know, maybe you're going to make a roast, maybe you're going to make some sort of dessert. You wouldn't want those mixed together at all. So, keeping things in chemical compartments is the job of membranes, because we want those reactions to happen in places where they happen at a efficient rate. So we want to concentrate them inside of things. We want them to happen with maybe an enzyme that speeds up the reaction or lowers the reaction energy. So controlling where things are is a really, really big deal. And that's what membranes are all about: letting things in, letting things out, and controlling chemical reactions. Okay, so here is your close-up of a cell membrane. This little cartoon here shows a lot of the components playing different roles in keeping things in and out and maintaining the environment that we are looking for. We're going to talk about each one of these, and just to point out to you, on the left side, these are the terms you should be able to define as we go through this. So, plasma membrane, it's not just these phospholipids that we'll talk about. It's lots of other things. It's transport proteins. It's these little glycolipids on here that we talked about when we talked about those when we did carbs as part of one of the functions of carbs. So there's a lot going on in the membrane that we're going to look at, and this is just an overview cartoon of that. So we'll get into each one of these individually. Okay, so breaking down membranes, right now we're going to concentrate on what a phospholipid is. All right? You've probably seen this maybe in a basic biology class. They make what's called a bilayer, which just means there's 2 layers. There's one, there's 2. So all of these lipids have 2 tails, so they kind of look like a lollipop with 2 sticks. And they have a hydrophobic and a hydrophilic region. I often draw them with a little memory tool. I'll put a heart at the top, and that heart is the hydrophilic, because phil means love, like trying to think of some Phil words. Philadelphia is the city of love. So, well, that's not exactly the Dr. Phil is not the exact word, same etymology. There's people who are audiophiles who love good sound equipment. So, and that end is this heart end is hydrophilic, and the tails, they're lipids, and lipids are hydrophobic because they have all those hydrogens on them. So we have this self-assembling layers. You know, if I dumped a bucket of Legos on the floor, they would not assemble into anything. But if I dumped a bucket of phospholipids into some water, they would self-assemble. So that's kind of one of the really cool features of these. Okay, so here we are kind of breaking this down on the molecular level. So you can see that you have a phosphate group right here. And you have your fatty acid chains, which make lipids. And you can see one of them is saturated. This one is saturated. And one of them is unsaturated. So it has this bend in it because there's a double bond right there. Okay, so we would see those are supposed to be carbons. There would be a double bond between two of the carbons on one of the phospholipid fatty acid tails. I'll never ask you to memorize this, but I might ask you at least what these two parts are and what they're made of. There's a phosphate group up here, and there are two fatty acids, which are lipids, and that's why they're called phospholipids. Okay, so let's look at what their structures, and again, I kind of went over on this side here. I always draw a little heart in here to make sure I remember that that's the hydrophilic, the water-loving end, and that I don't really have a memory trick for the hydrophobic tails, but I think if you get one, you can get the other. You've got some terms over here that I've gone over quite a lot, or quite in depth, so I think we're ready for that. All right, and just remember, self-assembling. Okay. So I kind of already talked about this, but when you put these in water, they make this bilayer, and the key word is in water. So the only place to hide from the water when you put them in water is for these guys to face each other. So the tails are kind of like protecting each other from the water. So these are all those little hearts, okay? And they are happy when they're facing water. And these guys here are kind of like, I don't want to get wet, okay? I want to stay dry. And so they face each other, and this all happens automatically when you put these into water, okay? So that the cool thing is these are self-assembling. If I went in here and disturbed this and poked through it, after things calm down, it would put itself back together again. That's really cool. So cells can be sort of disrupted, damaged, and they will repair themselves. Now, there are limits to that, of course, but I just want to give you that overview. All right. So this should start making sense to you. Over here is sort of a molecular view of this, and down here is sort of the cartoon view of this. And you basically have two very distinct areas. You have a hydrophobic interior. That's this area right here, or this area right here. And you have a hydrophilic exterior. Let's make that one green, which is going to be right here in this blue area. This is the hydrophilic exterior. Okay. So that makes it hard for some things to get through a membrane. It makes it easy for other things to get through a membrane. There are different factors that control that. Okay. So this is an extreme electron microscope view of this right here. And this surrounds the cytosol, the contents or the fluid that all the organelles are floating in. And this allows, again, internal chemistry that's going to be different from what the cell's environment is. No membrane, no cell. That's pretty easy to understand. Okay, so now let's take it a step further. Until now, all of our cartoons have been missing all these things. So, cell membranes have a very wide range of different proteins that are embedded. So, what would you guess might be some of the chemical properties of, say, this end of the protein versus the middle of the protein? What would you say might be, would this be a hydrophobic or hydrophilic area up at the top? That's going to be a hydrophilic area, and this area might be hydrophobic. So there's different ways we can structure these proteins so they point the right way, so they do the right job. These allow cells to do a lot of different functions, like let things in and out. Some of them are passive, some of them are active. You've probably heard of that, where active requires energy and we're usually going against the concentration gradient, and passive doesn't require energy and you're going from high to low concentration and things just kind of diffuse out without energy. So we'll get into that. So this is just kind of reiterating what I was telling you before. Membrane proteins can be hydrophilic and both or hydrophobic, and sometimes they'll have polar side groups, and this will determine how they arrange themselves within the membrane. That's sort of what I was sketching before, okay? So certain parts of this are going to have chemical properties that sort of anchor it in the right spot, facing the right direction. You wouldn't want this thing to turn 90 degrees and sort of get stuck in here, okay? We wouldn't want that at all. Some of the proteins are sort of peripheral proteins that only interact internally with these membrane proteins. And so these might connect to a membrane proteins, these guys down here, and change the state of that membrane protein to say, make this, activate, stop. And so these are kind of like traffic cops or red lights and green lights in some cases that tell these proteins to do their job or to change their job. All right. So here's an extensive list of different membrane protein types and functions, okay? So some of these proteins, I'm just kind of going to check mark my way down them. Hormonal binding sites. Your endocrine glands in your body make different hormones to regulate what's happening in your body. And there are different endocrine glands, like your pituitary gland, your thyroid gland, your adrenal glands, and these all have signaling jobs. So this is sort of like a slow signal versus nerves, which are a very, very fast signal. So your body might have a deficiency in one of these endocrine hormones, and we can treat you with drugs that will get those back to normal levels. A lot of people have hypothyroidism, where their thyroid glands in their neck don't make as much hormone as they should, and those people's metabolism slow way down. Okay. We have what are called immobilized enzymes. These are sites that catalyze reactions. So these are going to allow reactions maybe on the sort of inner or outer edge of the cell to happen at a lower energy. Okay. when we looked at cell membranes, when we did organelles, we had different things embedded into cells to allow them to sort of hold on to each other tightly. We have proteins that allow cells to identify things that are going on outside. So this might be a neurotransmitter. When a neurotransmitter crosses the gap between nerves, it'll bind with one of these proteins, and that signal will move down. It'll fire the neuron, which will propagate down the nerve. And then these ones are probably the ones you're more familiar with. We have transport proteins, which are either passive for no energy, or active transport, which are going to use ATP. Some of these are really, really cool. The sodium potassium one is one of the favorite ones in biology because it moves things in different directions using one ATP per cycle. Biologists love it. All right. So, everybody okay? Speed-wise? You need a little bit more on the last one?
Yes.
Okay. Oh, you guys don't have to copy that down, but I mean, if you want to really paraphrase these, that'd be great. I did, but it's in the CAMI. If you want just a straight up list of slides, I can throw that up there too. All right, if you're watching the video, my in-class people are working on paraphrasing this into their written notes, so just be patient with us or fast forward through this section. All right. Okay, I'm seeing pencils kind of slow down, so I'm going to go ahead and go unless somebody says, wait one more second. All right, everybody looks like they're caught up. Yeah, there's a lot of info on one slide. All right, just a few slides to go. So, when you think of a mosaic, mosaics are forms of art where people put little tile pieces together and make a pattern or even an actual sort of picture or representation of whatever. And so these are usually sort of glued or sort of cemented together, and once the mosaic is made, it's made, and you know, people are digging these, archaeologists find these that are thousands of years old and so on. And so these are not fluid. So they kind of throw this fluid mosaic as a term that means each one of these is sort of like a tile in the mosaic. But because it's fluid, they can sort of float around through here. So, you know, this phospholipid, like if I pick one of these particular phospholipids, over time it's going to flow through the membrane. And that goes for any other one as well. And that could depend on a lot of other factors. Now, we can do things to cells to kind of, we can do things to membranes to kind of minimize that if that's important to the function of the cell. So these guys randomly move around, and they can even sometimes move from, you know, this layer, inside layer to outside layer, and vice versa. So you can see things like that happen as well. Getting into some really small level changes. So you'd have to really study that closely at a really sort of micro level to see that. All right. Now there are certain proteins that end up usually sort of poking out of each end or one of the ends like an iceberg, okay? But cholesterol, so know what cholesterol, this is sort of a really important idea here. Cholesterol molecules, and these are found in animals, and cholesterol is hydrophobic. And so where's it going to be if it's hydrophobic? Well, it's going to be in the hydrophobic region. Now these help maintain the membrane. So if you have a membrane that you don't want to be as fluid, maybe a certain side of the cell has a really well-defined job, like in the case of not a cellular level membrane, but like a body membrane, maybe like the lining of your lung or your digestive system. You want those cells to be sort of sided really well. You have an environmental side and you have sort of an internal side. Those tissues are called epithelium if you're an anatomy person. All right, so what does this cholesterol do? It reduces membrane fluidity. So this is sort of like a key idea here. The number one thing you need to know, so by reducing the amount of movement in that fluid mosaic, we have a more tightly controlled membrane. So let me ask you this. Just so you know, which ones are cholesterol, A, B, or C? Just a little quiz. C, okay? Look, cholesterol is in the hydrophobic region. This is not in the hydrophobic region. So these little, hexagons that are part of the glycolipids, they can trick you a little bit. Cholesterols are on the internal side. So if you said C, you've got it. All right, and our last slide is just a review. So what's going on in this? Well, let's go back to that. What's going on here is we have this really new word here, probably, amphipathic, okay? I'm going to probably mispronounce this. Amphipathic. This means that it's hydrophobic on one side and hydrophilic on the other side. So that's one of the terms that you'll want to know, okay? You want to know why they form bilayers. They form bilayers because they either want to hide from the water or they want to point toward the water. Okay, so we looked at a list of membrane proteins. You should be able to tell me some of those functions. Okay, we looked at what cholesterol does in animal, specifically mammal cell membranes. And you should be able to draw or identify things that are going on in a diagram of a cell membrane. All right, we're going to go ahead and end this one. So this is 2.4, membranes.